This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 82, recorded on February 4th, 2015. Hello, everyone. I'm Vincent Racaniello, and with me here in the TWiP studio is Dickson Despommier.
Good morning, Vincent.
It is morning. How are you, Dickson?
I'm very well, thank you.
It's cold as heck out, isn't it?
Not today. No, it's been modified today. It's about
36
degrees right now.
Why are you still in
going
Fahrenheit?
Up to 70.
Can't deal with Celsius?
It's going up to 40 right now.
I started to pick
40.
on you already. No, it's
okay,
freezing.
No one cares.
It's 0 Celsius.
Yeah, so it's
here
30
in New York City.
32 degrees, but it's supposed to go up to around 40 degrees Fahrenheit by the afternoon.
It is going up to 2 degrees Celsius, which is not 40 degrees. Where do you get your weather from?
NPR.
Wrong. All they're good at is raising money.
National Public Radio.
Also with us today, right here in the TWiP studio, Daniel Griffin. Welcome back.
It's good to be back.
Third episode.
Yes. Third one's a charm.
Do you feel enriched?
I do. I feel like I'm learning something, and I'm very entertained that when we were discussing the weather, rather than the fact that some of us had been outside not very long ago, we didn't actually use that experience to gauge the temperature. We whipped out the iPhone. The iPhone says it's 0 degrees. I mean, if it had said it was 70, we all would have said it didn't feel like 70.
No, that's right.
Yeah, I mean, I knew it was warmer than it has been in the past few days,
right.
because in fact the roads are melting. Yesterday it was freezing. I know because I spent some time on the side of the road.
And did your iPhone say that it was cold? I mean, something objective.
So, Vincent, you want to share with us your day yesterday?
No, it's not a big deal. But you know what was
Have your
interesting?
adrenal glands come down yet?
I was very calm because what could I do?
I guess.
I broke down on the New Jersey Turnpike. It took a long time for the tow truck to come, and my car was off because it wouldn't run, and I didn't get cold. I was okay.
But that's a very dangerous situation for you.
Man, you know what?
Glad you survived it.
So right on the shoulder, these trucks. You don't care if I survive or not. Don't say
Vincent,
that.
who else would get to run all?
Well, it would be the end. It would be the end. But the trucks going by, vroom, vroom, the car moves each time. What am I going to do?
Exactly.
I go. And I was thinking Dickson will have to run TWiP.
Yeah, impossible.
We have some responses to last week's case study. Maybe you want to summarize and remind us what was that about?
Let me remind our listeners what the case was about. We were discussing a gentleman in his 50s who had been admitted to a hospital on Long Island. This gentleman was from eastern Long Island, and he came in with generalized symptoms, anemia, so his red blood cell count was low, a little bit of hepatitis. He was missing his spleen. We thought that might be relevant. He receives transfusions once a month, and as I mentioned, he's from eastern Long Island.
So I'm really sad because Long Island, well, that's where you're from, Long Island as well, but whenever you want to go somewhere, you have to go through New York. Unless you take the ferry, I guess.
Which we often do.
It's a pain, isn't it?
Anytime I can be on a boat, I'm happy. So even if it lengthens my trip, I'm driving Orient Point. I'm taking the ferry across.
I've done that too. That's quite an interesting trip.
But it is true. There are limited ways of getting off of. People forget it's an island, and there are limited ways of getting off. You pretty much have to go through New York City. You can take the Throgs Neck. But I think that drops you in the Bronx, right?
Yeah.
Something like that. So do I get a chance to guess at this first before we go to the listener picks?
You want to? I don't know. It's up to you. It's your show, Dixon, as
you're
always telling me.
It's our show.
It's our show.
This is a team effort here.
I would love to hear what our listeners.
Well, you're going to hear it.
Okay.
All right. Here's we have 5. Alan writes, "Dear Vincent, Dickson, and Daniel, I like your idea of a TWiP coffee mug prize, or maybe a mug discount. In this second case study you presented, the present symptoms are pretty vague, but the history is interesting. Also, since with this podcast you've given us license to hunt for zebras, particularly parasitic ones, I would want to rule out Rocky Mountain spotted fever, common in Long Island, but with the presence of regular blood transfusions and the splenomegaly in his history makes me think you were highlighting something more. I would want to also test for transfusion-associated malaria, dengue, or babesiosis. Although normally vanishingly rare as a transmission route in the U.S., the fact he's getting monthly transfusion moves this route to just very unlikely. I think there were several dozen transfusion-associated Babesia cases back in 2002, and I actually thought the U.S. was testing for it now. Otherwise, it's just opportunistic if around cattle and deer ticks. Between malaria and dengue, I doubt the patient would describe dengue as just fatigue, malaise, and stomach upset. I've had both, and dengue hurts worse than that. So if I had to guess, it might just be malaria, non-P. falciparum. I look forward to learning more from you three in such an entertaining forum. So to remind you, Alan is at the University of the Nations in Hawaii.
So, Alan, I think you missed a little point here, though, that the patient didn't have a spleen.
It's splenomegaly.
Yeah, so that's
okay.
It's a small glitch.
Kurt, right?
No, I think that's an interesting point. I was recently listening to these public health lectures where they're talking about how do you diagnose malaria when you're in a place where you don't necessarily have access to the technology that we're used to here in the U.S.
Right.
And one of the criteria that WHO puts in there is this febrile presentation and the ability to palpate a spleen.
Correct.
But I will say, having your spleen removed does not protect you from malaria.
No, nor does it protect you from a lot of other things either, but it certainly does protect you from splenomegaly.
That is true.
So this is a clear-cut case of reading a case or listening to a case fast and then making a judgment. The clinicians did not detect splenomegaly because this patient doesn't have a spleen.
Right.
But the fact is that he's the patient. You're going to read other diagnoses first.
Yeah, let's read a few. Kurt writes, "Hey, TWiP gang, loving the new format. This case sounds like a dead ringer for transfusion-acquired malaria. Test by PCR or slides, treat with anti-malarials." Short and sweet. Robin writes, Robin is our physician, retired, a case of transfusion-carried malaria. Anne writes, "Dear Daniel, Vincent, and Dickson, I have only recently discovered TWiP and the other Twix and have been avidly listening to both the current episodes and catching up on the earlier ones, if only I had a longer commute. As a veterinary pathologist, it has been a delight to hear about diseases and pathogens near and dear to my heart and training. Regarding the second clinical case presented by Daniel on January 24th, I considered Babesia microti and Anaplasma phagocytophilum high on my list of differentials, which included all of the diseases discussed on the show: babesiosis, RMSF, spotted fever, and malaria, plus anaplasmosis, ehrlichiosis, typhoid fever, and even Bartonellosis. Given the patient's regular history of blood transfusion, the issue of whether the infection was acquired through tick bites was less important than whether a classically tick-borne infection could be acquired through the blood supply. Similarly, although several of these are not caused by eukaryotic parasites, I figured they were still fair game since Rocky Mountain spotted fever has been thrown in the mix. A diagnosis of infection with Babesia microti is supported by the generalized and vague signs of illness, fever, history of asplenism, dark urine, mild elevation of liver enzymes, and geographic location of the patient. Similarly, these findings would also support a diagnosis of human granulocytic anaplasmosis, for which transmission through the blood supply has also been documented. Favoring consideration of HGA is the presence of thrombocytopenia as well as anemia. Were morulae observed on the blood smear? Although the presence and nature of a rash would not have been diagnostic in and of itself, it is a sign to be considered in several of these diseases. Looking forward to hearing the answer. Anne is head of pathology services out in Beaverton, Oregon.
We may want to get her on this show as a guest. She
and
sounds
our
like she's right on board.
Our last one is another Robin. "Dear Doctors of TWiP, long-time listener to all the shows, first time writing to you. My guess for the case presentation is babesiosis acquired via blood transfusion. That is what will be seen on the smear, I suspect. I very much enjoy the clinical application aspect of the show. I'm a registered nurse, 32 years, and amateur biologist. Dickson's anecdote regarding the effectiveness of freezing bear meat will make for great nerd cocktail conversation." Thank you, Dickson.
Do not eat the bear tartare.
As part of my job, I am responsible for monitoring and following approximately 50 patients per month for possible bloodstream infections, secondary to vascular access devices, for example, Picc lines. Picc? Do you know what that is?
Picc Lines. They're really a long indwelling IV. It goes in the antecubital, which is if everyone bend your arm and you've got your arm is caught there, so they like to draw blood. We'll feed up a catheter. And it's probably about, I'm going to use inches here. It's about 2 feet, about 24 inches. It feeds up, and it allows you to give medicines for prolonged periods of time.
I also recently diagnosed, thanks to TWiP, a colleague with dengue from travel history and symptoms. I use word diagnose in the loosest possible sense. Your shows have been a great source of learning for me. Thank you for sharing your time and talent with all of us. Kind regards, Robin. He has a last name. And he said, Vincent will be able to pronounce my last name correctly, I trust. I guess I can pronounce it since he said Rositani.
Rositani.
No. You didn't even come close.
No?
Rositani.
Rositani.
You don't have a good Italian accent.
I have no Italian accent.
All right, those are our 5 guesses.
Right.
And take it away, Daniel.
So wait, I get a guess.
Yes.
Number 6.
You want to guess?
So the fact that he lives on Long Island and it is associated with endemicity for the organisms that were ticked off by the veterinarian doesn't necessarily mean that the person acquired the disease from someone else that lives on Long Island. I don't know where that blood came from. So if he gets regular blood transfusions, they could come from anywhere. So the fact that he lives on Long Island doesn't necessarily mean that he has a Long Island-based disease. And that was my logical thinking on this one. Since we want to use Occam's razor to look for the simplest explanation for a complicated situation first, and then eliminate that, and then go on, I would side with those that picked malaria, transfusion malaria, as the reason why this person is in the hospital.
Really?
I would do that.
Okay.
And it would be either Plasmodium vivax or Plasmodium ovale.
Why would it be those
Because
two?
they're long-lived in the blood. They tend to relapse, and people tend to forget that they had malaria, in fact, and then they go in and give blood, and the next thing you know, there's another one, too, Plasmodium malariae, which is recrudescent, and you can get that one for 30 years out. I mean, it's really crazy. So people do give that one also for transfusion, but the most common one is Plasmodium vivax.
Okay.
Again, I'm always impressed with the level of sophistication of our listeners and Dickson. And I think the consensus, what we did last time, is we asked, well, what test will be the next test? And I think we're getting from all parties, they'd love to see a blood smear.
I would love to see a blood smear.
And they would like some serologies. I will start with the blood smear because the blood smear is easy. You can do it right away.
Sure.
You can actually go look yourself. And in this case, this gentleman, I actually went looked myself.
Oh, you did this diagnosis yourself?
Which is great.
Cool.
Well,
I mean, you know how to use a microscope?
We're actually
You're a doctor and you know how to use a microscope. It's not a joke.
Not only that, but they let you in, and the people in the lab are very nice to you.
Yes.
I think it's like when you travel to a country and they speak Spanish and you know a few words.
Sure.
Actually coming down to the lab and wanting to see the slide as
You
opposed
bet,
to just
you
read
bet.
the report.
It's like going to a restaurant and going back into the kitchen to see how the chef behaves.
Yes, but again, you can't actually act on that knowledge because there's all kinds of certification.
That's right. Okay, so
and appropriately so.
We're on the edge of our chairs here.
Everyone's on their chair, and the blood smear gave us the diagnosis.
Ow! You're going to just show it to me first?
I want to show it to Dickson, and he can describe what we saw in 10% of the red blood cells.
All right, these are very interesting organisms here. These are malaria-like in their appearance, but there is a dead giveaway here. It's almost pathognomonic for the correct answer, which is called a Maltese Cross. So it does turn out to be Babesia microti.
Yes,
because if it was Plasmodium falciparum, then you would have seen some stages of the infection, which would have caused an enlargement of the red cells. So that you don't see an enlarged red cell here. And the appearance of these rings, which you would mistake for malaria parasites, they look double-dotted for the most part. Now, granted that Plasmodium falciparum can look like this too, but Plasmodium falciparum is not usually blood transfused because it's an acute infection that's usually treated, and there are no residual liver stages to exacerbate later on. So if you had to make a choice here, this would be somebody from Long Island that didn't know they had Babesia, gave blood, and this poor fella got it instead. But there's a big...
Why would it have to be blood transfusion associated, though?
Well, it wouldn't have to be. Of course, he could have caught it from a tick.
Yeah.
He could have caught this disease from a tick, and the transfusions are just red herrings.
Now, one of Anne had said where morulae observed. Is that what?
No. So
no,
morulae is the pathognomonic or the characteristic finding you see in anaplasma infections.
Right.
What Dickson was looking at, I showed him a picture here, is a number of red blood cells all of fairly uniform size, and ring forms. They actually look like a diamond ring. A thin part of the ring and then a thick part, or a signet ring.
That's right.
And then the other characteristic part is called this Maltese Cross, or we say tetrad forms. And it has to do with the biology of the organism. When it divides into 4. And you can actually... They're hard to find, in all honesty. You got to spend time. But, it's actually fun.
But you said 10% of the red cells were infected.
Now, in this person, 10% of the red blood cells had rings. This is on a thin smear, so it's very obvious.
So he's an acute phase infection here.
And you're able to find these tetrad forms.
So this guy doesn't have a spleen.
And I think that's important in a lot of these cases. They're a healthy... 99% of the person will get exposed to babesiosis, will be fine. But people who have really characteristic no spleen or some other immune challenge are not able to control the infection. But let me also get to the point, did this man get it from the tick or the blood transfusion? And when I was here at Columbia, I first came, they do these rounds where they present interesting cases, and they presented a Babesia case. And living in Long Island and seeing so many as they present, I'm like, oh, that's Babesia. But if you're not from the endemic area, it is much more interesting. I'm sure some of our cases when we presented, the case previously, if you're from Bangladesh, you're like, oh, it's that. You see that all the time. That's not that interesting. And areas where they see malaria, when we talk about malaria, they're like, really? That's like
these are not zebras in those countries. Those are horses.
So this is not a zebra in eastern Long Island, in Nantucket, in Martha's Vineyard.
That's correct.
And so that's helpful. The other part is this infection can have a long incubation time. So it's not clear clinically at this point whether or not he got this from the transfusions. And we don't screen the blood supply for Babesia.
That's right.
We do for Chagas since the late, 2007 we started doing that.
And we don't screen for malaria either.
We don't screen for malaria.
We ask you if you've ever had it.
Yeah, we ask you screening questions, and that's
and you might say yes or no, depending. And by the way, we stopped paying people to give blood for that reason, because people would lie and say they'd never had something, and then they could give blood and get paid for it, and that really confounded the issues.
I see.
True. So in this case, this person could have gotten a tick bite back in, let's say, late summer, late fall. Here we are 6-7 months later. He also could have gotten it from the transfusion. The incubation time from the time you're exposed to the time you're sick can be as short as a week or two, but it also can be months. And a challenge is treating someone who doesn't have an intact immune system. I was
going to say, what are you going to do about what happened to this man?
This man actually required prolonged treatment.
With what?
I wrote the things down of what we do. And there's a couple. I don't know how excited our readers are about all the choices of what we did in this man, but we did. We initially started off with IV clindamycin because, as we saw, 10%. There also is the other issue about, do you go ahead and do exchange transfusions when it's that much? This person actually got better without resorting to exchange transfusions.
That's remarkable. Because in the old days, when this first hit the stands in terms of, look what's happening out on Nantucket, because that's where this first was detected, if you didn't have a spleen, you thought you were going to die from this, and they usually did.
True.
So it could have been fatal. So clindamycin, which is not a typical anti-malarial drug, it inhibits protein synthesis, if I recall correctly. The mode of action is, I know Dr. Vincent Racaniello is going to look this up for us.
You'll look it up. And we also, you use a second drug when you're treating, similar to malaria.
Because that's pretty slow acting, right?
The clindamycin?
Yeah. Because it's given in conjunction with normal drug-resistant malaria as a way of slowing it down so that eventually you can put in other, like artemisinin or something else that would interfere with the biology of the parasite. But in Babesia's case, the anti-malarial drugs, they just don't work.
Yeah. I mean, in this case, depending upon the severity, depending on the percent of Babesia, there are oral options where you might use a Tovaquin, or maybe people know as Mepron, and a Zithro, that wonderful Z-Pack. Or when they're above a certain level, the idea is you'll use a clindamycin plus a quinine. Imagine that.
Potent.
Yeah.
You wanted to know the mechanism of action, Dickson?
Yes, I did.
Yeah, it inhibits protein synthesis.
At what level?
Ribosomes.
That's exactly what I thought, but I didn't want to say it because if I was wrong, then it'd say, aha, he's wrong again.
It's used for anaerobic bacteria, right?
Yep. Yeah. Yeah.
It is. And life cycle. Did you want to discuss the life cycle?
I
The
was
life
gonna
cycle
ask
you. Go for it.
is fascinating.
No, do it.
And some of our listeners who brought up other tick-borne infections,
Right.
were right on, because what his Occam's razor was great in the good old days when everyone only had one infection and ticks only carried one pathogen.
That's right.
But Occam lived 600 years
Since
ago.
I don't shave anymore, a razor blade is kind of a foreign thing to me anyway.
And maybe in Occam's time there was only one disease.
Yeah,
And but nowadays the ticks will carry many diseases.
That's true.
So a lot of what we say about Babesia relates to Lyme disease. That a lot of people know
right.
about, and it's the same tick. It's the same. We call it the deer tick. It's a
Ixodes.
terrible name. Why do we call
know.
it the deer tick? I wish we had called it the white-footed mouse tick. Right? Can we do that?
We could, but the adult tick actually lives on deer.
And I'm going to talk about it because I think that's a great part of this. You end up acquiring this from ticks.
You do.
Summer is kind of, particularly the end of the summer is.
So there's a seasonality.
There's a seasonality
that relates
to
to
this.
the ticks, right?
And it clusters because most of the incubation is not 6-7 months. Most
Right.
of it is a matter of weeks.
Right.
You get bit by these tiny nymph forms, which, as we discussed before, about the size of a poppy seed.
Indeed.
And you can actually get infection from these.
Right.
Now, where's the deer? We call it the deer tick. And
Long Island? Are you kidding? There are more deer on Long Island than there are people that live out in Montauk. I know this for a fact.
And they've done these wonderful, we'll call them wonderful, they've done these deer control programs with this idea that if we only kill 99 out of 100 deer, that will have less of a problem. And for some reason, when they do that, it really doesn't have a major impact.
No, because
and why is that? It's a deer tick. What's going on here?
yeah, but other deer come into the area, though. This is a problem. I think if you could eliminate deer altogether, this would disappear.
Well, I would say if you could get 100%, this is my little, maybe the readers will
Yeah,
enjoy now how I think of it here.
Yes.
The real important part of the deer in the life cycle is that's where the female and the male ticks meet. So I think of the deer as the singles bar for ticks.
This is true.
And
This
let's
is
say
true.
you have a town and there are 4 bars in that town and you shut down 3 of them. Are people going to stop drinking and going out? No, they all go to the last one.
This is true.
And so if you're going to control the deer, you're going to have to get rid of them all. But the mice, if you can
that's
control
right.
the mice, that's an important part of the life
Much
cycle.
more difficult, though, because their mice are more prolific. They're more cryptic in terms of where they live. They have fewer predators in populated areas because they've eliminated all the coyotes, although they did find one not in Central Park, but in New York City.
No, there was
just
a
recently.
Central Park coyote within the last few weeks.
That
I heard-
was Central
yes.
Park? Okay,
I
fine.
heard.
So that's the second one that's found its way into New York. Looking for a singles bar, no doubt. But the point is that Daniel is raising is that what is the definitive host for the tick?
Is it the mouse?
Yeah, no, it's not the mouse. It's the deer. It is the deer.
Really?
The adult ticks live on the deer. The larval ticks feed on mice, and once they take a blood meal, they're good for the next stage of their development, so they drop off the most mice. But in the meantime, they've given the mice the infection too, of course, which is why it's called microti. And the other thing is that this is an unusual protozoan infection of ticks,
Right.
because in mosquitoes, if this is a malaria parasite, it doesn't vertically transfer from the egg to the larva to the pupa to the adult. Whereas in ticks, this is an incomplete metamorphosis life cycle. So it's an egg and a nymphal tick and a bigger nymph and a bigger nymph, and finally you've got an adult. And the infection does vertically transfer, along with rickettsia and all these other viral infections too. So ticks are the best vectors in this case for this particular. Thank goodness that ticks don't transmit malaria, because if they did, you'd have the same problem with malaria that you do out on Long Island with Babesia right now, except all over the country.
That's a great, to compare this to the malaria,
Yeah.
where the ticks are born. The little nymphs are born with the Babesia, with the Lyme disease.
And that's how this guy got it. He didn't get bitten by an adult tick, probably. He got bitten by a larval tick.
Right, you can hang out with the deer. They're fine. It's little.
That's exactly right.
But the ticks, as Dickson mentions here, to go through each step of their life cycle, they require a blood meal. Whereas we discussed with the mosquitoes, the mosquito requires a blood meal for egg production.
This is correct.
And most 99% of mosquitoes only bite once.
You got it. Not only that, they're short-lived. So mosquitoes can live maybe 3-4 months at most as an adult. Ticks, I know this sounds ludicrous, but the old literature has someone keeping an adult tick that had been fed once for over 50 years, and it was still alive at the end of the 50 years.
That is a long time.
That's a very long time. So imagine all these adult ticks as reservoirs for this infection. They don't go away. They stick around a long time. So this is a very interesting case for many reasons.
So I was looking at the CDC page with that life cycle. They had a link to an article of transfusion-associated babesiosis in the U.S.
Right.
So apparently the first such case was described 1979. And in this paper, they describe 159 transfusion-associated cases in 19 states, although most of them were in the 7 main microti endemic states. And they say at the end, donor screening strategies that mitigate risk for transmission are needed. Babesiosis should be included in the differential diagnosis of unexplained post-transfusion hemolytic anemia or fever, regardless of season or U.S. reason. Is that a reasonable?
Yeah.
I think that all sounds reasonable. And I would say the case that they presented here at Columbia as this challenge, they actually went to the extent of tracing down where did he get the blood, testing the blood, in that case showing that it was from the transfusion.
Right.
So people will go.
Sure.
And this is as a growing problem,
So it must also be stated as a historical footnote that the tick was the first arthropod vector ever identified to transmit diseases.
Mm-hmm.
And this
guess
is great.
who did it? His name was Theobald Smith.
Theobald Smith, who's got a place named
There's a
after him
whole
at
building
Rockefeller.
named after him at Rockefeller.
Theobald
because that's
Smith
where he
Hall.
worked. He worked for the Rockefeller.
Did you work in Smith Hall?
I did, as a matter of fact. I worked on the 3rd floor in Smith Hall with the William Traeger group.
Yeah. So that's where my wife worked when she was with George Cross because he was given Traeger's old space.
Look at that.
They kicked him out. One more thing, Dickson.
Yes.
The rodent host of the tick that has babesia is the white-footed mouse.
His last name is?
Paramyscus leucopus.
Leucopus, the white-footed mouse.
So Paramyscus maniculatus is the host of hantaviruses.
But I'll bet you these can carry it too.
There have been a few cases of hantaviruses out here, right? I think one on Long Island, Daniel, isn't that right?
Yeah.
I saw a number of hantaviruses because, as maybe our listeners remember, I spent a lot of time out west and was in Utah when we were doing the ribavirin trials to see if that would help with hantavirus. And I saw several cases. I remember a mother and daughter, and you worried more about the women. They would go up to clean the cabins.
Of course.
And the men, I don't know what the men were doing. I'm not sure why they don't get to clean the cabins.
At the local bar.
And they would go up in early spring to the mountains.
Yeah.
And they would sweep it out, and they would aerosolize. And then, you'd see.
Yeah.
I had a patient recently here who had just such a wonderful story, but he didn't have hantavirus.
So the one in New York was in 2012. It was upstate New York. It was a guy who was bitten by a rodent while camping in a lean-to shelter in the Adirondacks. It was laboratory confirmed.
Interesting.
So we have him here. All right. Now, when do you have to leave, Daniel?
I have to leave at noon. We have a...
All right. Let's get into our paper, and then we'll do some... we have to do the other case study, of course. So we need to leave time for that. I'm going to let you take the lead on this paper, Daniel. Is that okay?
He selected it.
I was hoping to give the lead to Dickson, but that's fine.
You really want to give it to Dickson?
I will jump in, and we
will...
No,
it's not permitted to give him an immunological paper lead.
Well, how about if I start off with a preface?
Yeah, sure.
Preface of this paper.
Why don't you read the name and
going to,
stuff, Dickson, Despommier?
I'll try to keep my mouth close to the microphone while I read, but it's... long-distance reading is not possible for us. So the title of this article that Daniel selected from PLOS ONE is called Secreted Proteins from the Helminth Fasciola hepatica Inhibit the Initiation of Autoreactive T-cell Responses and Prevent Diabetes in the Nod Mouse. And the authors are Maria Lund, Branwyn O'Brien, Andrew Hutchinson, Mark Robinson, Ann Simpson, John Dalton, and Sheila Donnelly. All good Irish names. And indeed, some of these people are from Ireland. They're from the School of Medicine and Biomedical Sciences in Sydney, New South Wales, Australia. from Queen's University Belfast, Northern Ireland, Institute of Parasitology, McDonald Campus, McGill University, and the I3 Institute, University of Technology, Sydney, University of New South Wales, and one flyer in this one was from the University of Paris, Descartes, France. So it's interesting because this parasite that they're going to discuss in terms of its ability to alter the immune response of a host that suffers from an immunologically based disease, autoimmune disease, that we would refer to as type 1 diabetes, all these people live in Commonwealth countries except the person from France. And in each of those countries, this parasite dominates the scene with regards to sheep. This is an endemic infection in Ireland, in Australia, in England, and in a lot of other places where sheep herding and sheep raising and farming is the deal. So they're looking at a parasite, which is a cestode, which lives in the liver. Did I say cestode? I meant trematode. I misspoke. It's a trematode that lives in the liver as an adult and creates tunnels that are so large that you can actually see them on ultrasound. And they cause horrible problems in sheep. They I wouldn't say they decimate because that means 1 in 10. They are worse than that. Their larva insists as an environmentally resistant stage on watercress in aquatic environments because all of the trematodes require snails in order to reproduce. So the sheep living along the side on a farm goes down to the water's edge to drink and at the same time takes a mouthful of watercress, chews it up. This little cyst then excists, and it has a weird life cycle that it actually penetrates the gut wall of the small intestine and migrates in the peritoneal cavity through the capsule of glycine, which is the membrane that holds the liver in place, and drills itself down into the liver tissue, which is remarkable because it requires 2 of these parasites to mate successfully. They're hermaphroditic, but they don't mate with themselves. They require another parasite in order to mate with them. And so each one does this, and somehow they find each other in the liver. Yeah, so there must be a lot of pheromones involved in all this stuff, too.
They find each other delightful.
Well, we had a show on that, actually, in the tall grass. That's right. So we'll go back to that show. So what we've got here is a parasite which causes a lot of economic damage to sheep. The farmers that raise these sheep, and not only that, sometimes, of course, the farmers acquire this infection as well. So, but it's not as common as Echinococcus granulosus, which is another sheep parasite that we'll talk about. We have talked about. But here they're using it as a tool. They're using it as a tool to investigate whether or not this particular helminth has the ability to immunosuppress autoimmune diseases. That's an interesting concept that has arisen out of the literature over the last 10 years, using parasites to ameliorate the effects of Crohn's disease, irritable bowel syndrome, and now they've added another long list of these. And in fact, I downloaded a table from a review article in which helminths have been used, and we'll get to that later in the article, have been used to see whether or not they've had an effect on these autoimmune infections. Or diseases,
Dickson,
I should
last
say.
week or last TWiP, we talked about
We
immunosuppression in water buffaloes.
Yeah, that's exactly right. So now, I think Daniel is right on point in selecting this article because it's got tons of immunology in it.
Just right for you, Dickson.
It's got tons of jargon and lots of abbreviated words like PLD this and PD fact and TGF beta thing. I'm being a little facetious here because I do know what a lot of this meant. I did have to look up.
No, you're not being facetious at all.
PD-L1. Do you know what PD-L1 is?
PD-L1?
That's right. Do you know what that stands for? Come on, Vincent.
No.
Now we're going to have a game here.
PD.
PD.
Is this pancreas-derived L1?
It's called programmed death.
Yeah. Okay, Dickson.
Death.
We need to let Daniel talk because
so now take it, Daniel.
If you want to just throw out acronyms and think you're going to trip me up, find a better way. Find a better way.
I'm sure you can beat me at that game, but
no, it's not a game. How about TGF, Dickson?
That's transforming growth factor.
How about PVR?
That's parvovirus retro.
Okay, so they get the point. Daniel, what's this about?
All right. And TLA, do you know what TLA is?
TLA.
TLA, 3-letter acronyms. All right. So let's actually get to the article.
Please.
ASAP.
Okay. Yes. That was 4.
That's true. Well,
that was an FLA. Come on, let's stick with the TLA.
Vincent already knows I
What
can't
about
count.
NOD? In the title, it's NOD, Daniel. What
Yes, the
what does that stand for?
non-obese diabetic mouse, and I'm going to discuss that.
Okay.
I want it now. This is a very dense article. I have
Very
to
dense.
say, and I don't want to turn this into a science journal club for the elites who actually know every TLA.
You're going to give us the overview.
We're going to
right?
get the overview.
Yeah.
We're going to discuss
bet.
a little bit of why this is important. Now, the reason I wanted to discuss this was I felt a little bad last time when we discussed the immunology that was going on. I said, "Oh, it's very complicated." Well, I'm not saying, "Oh, it's so complicated and no one can understand." No, it's complex. There's complexities here, but we have a rather sophisticated audience. And Dickson, and I'm sure all of them can understand this.
And we're all good teachers. So good teachers can take a complex situation and make it understandable for the common audiences. They can do that, and that's what we're in the business of doing.
Well, I'm not saying our audience is common. I actually
No,
think
some of those listeners out there have never had any science backgrounds, but they love listening to science. We've had that
yes.
as many
Sure.
comments.
We
So
have a
that's what
broad
we're going to
range,
do
yeah.
today.
Therefore, I will address all of our listeners.
Would you please?
The great thing here is they're capitalizing on this concept that I know Dickson champions, the idea of turning swords into plowshares.
Right.
Taking a pathogenic organism, a disease-causing organism, and trying to exploit its tools for health, for
Here.
improvement of health.
Here.
And I think that this is a great paper where they demonstrate that, and we'll go through. They're talking about type 1 diabetes, and
Right.
we'll make the type 1 versus type 2 diabetes. It's probably important that we point out what's the distinction.
Right.
A long time ago, people probably are familiar with the insulin-dependent, insulin-non-dependent, and it was replaced because sometimes people with type 2 diabetes require insulin, and they're actually dependent on the insulin.
Yeah.
So you don't want to change the disease that you have just because you now require a new medicine.
Right.
And the difference being that type 1 diabetes is an autoimmune disease where we have the cells that secrete insulin, the pancreatic beta cells, destroyed by our own immune system.
Indeed.
When does
And-
this happen? Early or later in life, typically?
Well, usually early in life.
Okay.
This is usually before you're 20, where the type 2 diabetes, you're often producing plenty of insulin for a lot of the disease, but you become resistant to that insulin. And then by the time you're diagnosed, then you start to lose 90% of
When
your
you say resistant, you mean it's in the circulation, but it doesn't get into the cells to facilitate the uptake of glucose?
The insulin's actually working on a receptor on the surface of the cell, and
Okay.
despite the adequate amount or even a high level of insulin, the downstream action, the glucose transport, is not
Okay.
affected.
Okay. Which is somehow linked to obesity.
In 90% of type 2 diabetics in the U.S., obesity is an issue. That changes. I mean, we probably have an audience interested in the whole world. That changes in places like India and China, which now have a raw number of more diabetics than we have in the U.S. And it may actually have to do with malnutrition in utero. It's very interesting work on that. Type 1 diabetes is different, so we're going to keep that separate. This is, you have an autoimmune problem that destroys your ability to make insulin, destroys the cells that make them.
Is it specific to insulin-producing cells, or do you?
It is, actually. It's probably, we think, an antibody that is specific for those cells, for those pancreatic islet cells, destroys them, and they're not there. And there's an interesting sort of dip we see where initially you're under stress and the person, oh my gosh, this person has diabetes, and they get a little better. They call the honeymoon period. And that's because they just have a few percent of these cells remaining. And then the next stress, when you need 100% of your insulin, it's not there, then the person ends up, as we say, dependent on insulin.
So as an aside, isn't this sort of a crossover concept with regards to TWiP? Because I know in TWiP we've discussed the possibility, at least, that the autoimmune disease trigger was a viral infection that a child acquired, and we don't know which viral infection it is.
It could be.
It could be. That's the point.
Yeah.
That was the leading hypothesis for many years. I don't know. Has there been any improvement of that? Dan, do you know of any new data that suggests that we're closer to understanding which virus it might have been?
I don't think
If any?
I don't think we know what the trigger is. but in this paper we'll discuss a little bit about our more sophisticated understanding of the immune system that drives this.
Okay.
So let us.
Okay.
There's a bunch of terminology in here that we're going to try to work through. One of the first is Vincent brought up they're doing this on NOD mice, the non-obese diabetic mouse, the NOD mouse, and they're only doing it on female mice. They only pick female mice. What's up with that? Why female mice? There's several reasons why you might want to work with female mice. One is it is a little bit cheaper because they can live together. Guys, we know guys. And male mice are just like male
cheaper,
humans.
that's one good reason.
They got to keep them in their own cages, otherwise they beat each other up. They're not very nice.
Mice are fairly cheap, though.
The other that's
not mouse care.
Mouse care. You can
That's the
buying. It's
listen. That was half of my budget for my NIH grants.
Yeah.
Believe me, I know all about that one.
It's like buying a boat. The purchase price is not what kills you.
It's about maintenance.
And the same, every single male
right.
mouse. Okay. And the other is that this is a wonderful model for diabetes. 60 to 80% of the female NOD mice get diabetes. Only maybe a quarter of the males. Which, again, you'd have to use more mice if you're going to. And that's interesting. Why is that?
So the estrous cycle does not interfere with these data at all? Because they do in certain parasitic infections. So the male mice are favored over females because they're more homogeneous in terms of their hormonal backgrounds. So there is a difference there between males and females. And traditionally, if you look at the literature for parasitic infection research, it's the male animal that's been picked out as the result. So this is quite different, and I'm intrigued.
So that is this only apply to females?
Right. Another good question.
You would have to do it again in the male and decide is there some.
Of course.
Now, what is the plowshare? What are they doing? They're actually taking the fasciole hepatica. They're getting it from the sheep.
Right. Which is no problem for these people.
They're throwing it in a dish,
right.
And then they're collecting the excreted products. They call it
secreted. ES.
EX.
Yes. Excretion, secretion.
Yeah,
they're not sure which is which, so they call it both.
They do. They say secrete and/or.
No, I worked on that acronym for many years for Racaniello.
So you helped them with the F.
No. I inherited that term from an older literature, but it's the way they refer to it. They're not sure if they're excreting something or secreting something that's necessary for their life, but they just throw it all together and say ES.
And I think that's fine. We're the products that end up released.
Yeah, exactly.
That's what we're after.
And later on in the article, you'll see they comment about the complexity of that solution, which is very interesting.
And what they're going to talk about here, we talked about last time this idea of shifting this TH1 versus TH2. But what these authors are going to introduce is the concept of active immune suppression. The suppressive cells, suppressive B cells, suppressive T. Why don't we call them regulatory? And there's a reason why we call them regulatory. It is a very contentious history.
Indeed.
But the idea that there are T regulatory cells, and I like that because we're not suppressing the immune system, we're regulating it.
Exactly.
B regulatory cells, T and even these macrophage regulatory cells get introduced here.
Right. The M2 cells, so to speak.
Yes.
See, I used to do work in this area. It's a dim memory, I agree, but it's coming back. Every time you read an article like this, a lot of these terms come back.
You don't read articles very often, so.
I wouldn't say that. I wouldn't say that at all.
You read the abstract?
I read the articles. I read.
He read this one twice.
He did.
He read this one twice.
No, I read it more than twice. Actually,
I read it once like 2 weeks ago, I'm relying on my notes. But to get back to the article. So this will be a great article for establishing, because
you have 15 minutes.
Okay, cool. So we'll come back, but we're going to come back to these themes again and again.
We will.
We're going to be talking about TH1, which is the gamma interferon, we remember from our last episode.
Right.
We're going to be talking about TH2,
Yep. which
I mentioned. IL-4, IL-6, I think both of those are important, as we realize in this paper. And then we're also going to be introducing the concept of regulatory B and T cells.
Indeed.
And what they're showing in this paper are several things. But if you actually give these mice, it's an intraperitoneal injection, I think?
Yes,
an intraperitoneal injection
10
of
micrograms per injection.
That you're actually going to induce regulatory cells.
Remarkable.
And, the end result, you're actually going to be protecting these mice from developing diabetes.
Exactly.
So what are the numbers? So you said what percent
84%.
spontaneously?
we're protected.
So as we talked about, if you do nothing, 60 to 80% of the females, we're using females, are going to go on to develop diabetes.
Okay.
And we're going to drop that by about 85%.
Wow.
Right.
So it's pretty big.
Huge. It's a great result. It's not statistically marginal.
And this is achieved by injecting secreted/excreted stuff, proteins,
adult
other things from
trematode.
from these cestodes or trematodes.
It's a trematode. It's a trematode.
And this could be protein, lipid, carbohydrate. We don't know what's in this.
We don't know what's in there, but what is critical is they're injecting it early in life.
Yeah.
When they think
right,
that there's
that's right.
some sort of a opportune
induction,
time to
an induction phase.
before this
That's
gets
right.
inducted.
That's
So
right.
you don't have to keep taking this. This is a medicine you take your whole life
Right.
potentially. This
Right.
is something you step in during a high risk time for developing this autoimmune response, and then the mice end up doing well.
Remarkable.
You would have to give this early in people if you wanted to use it for that. Then,
That would
right?
be
Well,
that would
yeah.
be the challenge. For
How do you
identifying,
identify them? Yeah.
Yeah, who is at risk? You know,
So you give it to everybody.
No, you're not going to give this to
No,
everybody.
I
No way.
I don't think you
But
want
there's
to.
no genetics.
No,
There's
you-
no genetics in humans
I-
to suggest that there's a group that you could target.
There has to be.
I think eventually there will.
Has to be.
And it is a challenge
There
because
doesn't have to be. Why do you say that? There
Because we
has
have
to be.
3.2
I
billion
would argue
base
against
pairs.
that tremendously.
Well, then you should go home,
especially if it's
because-
due to an autoimmune disease that's
Of course,
induced
it's
by
genetic.
a viral infection.
Of course, it's genetic. It may be a virus, but it's genetically predisposed, Dickson.
You're predisposed to that virus, do you think?
No, I didn't say that. The infection predisposes you to something that you have encoded, for sure. You know why you get... Never mind. It is definitely genetic. I will bet you your fortune.
Let's
Let's
Good.
let
You're not going
Daniel...
to win much on this one.
Let's let Daniel...
I'm with Vincent on this. It's science. Just give us time. We'll figure it out.
Don't take it personally.
We'll figure-
But you're wrong. We made Dickson sad.
No,
You're just not listening, Vincent. I said that there's no genetics to the viral infection.
Not that
There may
we-
be a genetics to this.
There are genetics to viral susceptibility as well.
Yeah, but this is pretty broad-based, considering how broad-based
Okay.
the type 1 diabetes is in humans. That's all I'm saying.
Okay.
What fraction of the population has type 1 diabetes?
Now, type 1 diabetes is a lot less common than type 2. Type 2, they're saying before too long it's going to be the majority of our population. I mean, it's
Wow.
frightening.
That is frightening.
I'd have
Because
to
obesity is that
Google,
way.
because here you're talking in single-digit type
Right.
1 diabetes, much less common. So it really is. It gets lumped in together. We say, oh, we're doing diabetes research. But they're really two different diseases.
You're right. And we have a center for that right across the street, by the way, so we could actually call up an expert and ask them the details about all of this. But I don't think there's a familial pattern to diabetes type 1. That's what they haven't found.
It's harder to, yeah, there's a stronger, or at least at our current state of knowledge, there's a stronger genetic component, and maybe we'll say coming from the moms for type 2 diabetes.
Right.
But what is amazing, as we were saying before, is we used to say, oh, this is just a problem of excess. We eat too much, and if we can only lose weight. But as I was pointing out, this is a growing problem in India and China, areas with large malnutrition
So these kinds of results offer some glimmer of hope of treating an already induced disease.
After you have
reverse
it, Daniel, after you already have type 1, would this help? Did they do that in these mice?
No, and as we talked about, once those cells are gone, they're gone. But how could this help? Now what they're starting to work on is transplants, giving people eyelid cell transplants. And what is key is, can we convince the immune system not to destroy those?
Right.
And here we're seeing that we may be able to
That's right.
modulate our regulatory cells. And
Right.
modulating our regulatory cells is a way of not rejecting transplants.
Sure.
So instead of in these incredibly toxic drugs that we now
Yeah,
use,
they
where
give 'em
how do we
cyclosporin.
modulate our immune system? By wiping it out. That's
Exactly.
not modulating the immune system, but that's the current state of transplant. Let's
Yeah.
just wipe it out. Let's cripple it.
Sure.
We really don't want to do that. We want to induce active tolerance. We want our immune system to go ahead and see what's there and respond by tolerating it, just as we tolerate the rest of our body. And hopefully this is one step in that direction.
Indeed. Are you excited as a clinician to see these kinds of results?
Oh, I love articles like this because I see us still being in the dark ages in a certain way
is true.
with the toxic drugs, and then I get called because this person has no immune system, so they can tolerate their kidney or their liver or whatever organ, and now they've got some horrible infection because of that. And hopefully one day we will not suppress the immune system. We will modulate it.
We will manipulate it.
Modulate.
Okay.
Manipulation is this horrible kind of... I don't want to be manipulated, but if you modulate me, I'm not sure if that's bad.
Can you tell us what they've learned about the mechanistic aspects of this, putting this extract in? What is it actually doing? Do they know what cell types are being affected?
They do, and as we talked about, they are actually increasing the number of these IL-10 secreting B regulatory cells. They're increasing the CD25 FOX3 positive T regulatory cells. And then what may be new to a lot of folks is the macrophage regulatory cells, and their gene expression is also being increased. So we know what's being modulated. The really interesting thing is what's in the mystery broth.
Yeah.
What is it in
But
this?
do we know why that modulatory effect would suppress the development of type 1 diabetes? We probably don't know exactly what's going on, right?
No, not
It's just
not
that it makes
exactly.
sense that these regulatory populations would be the ones affected, right?
True.
But it's a cell contact killing probably that results in the death of the pancreatic cells, because that's a type 1 mediated response.
But you said that there are antibodies that are probably
I mean, there's a
involved, right?
combination, yeah. There's the antibodies that are targeting, but as we're seeing here, there probably is a cytotoxic. The cells are actually destroying
So
the
what
cells as
what
well.
antibody types are regulated here? Because they did mention that too.
They did do that, and I made some
Yeah,
notes about
IgG1
they
and
talk about
IgG2A.
IgG1, and I, even though I'm an immunologist and a B cell person,
Did you think much of that finding?
we think of IgG2 as a Th1 response and IgG1 as a Th2, at least in
There
the
you go.
mouse, and this was
There you
mouse
go.
stuff.
So it was consistent with the
It was
hypothesis.
consistent with the idea that in addition to regulation, you're also getting some change in the
Right.
focus of the immune response.
That's right. So I had a really interesting quandary that I found myself in. I didn't recognize the term PD. They investigated PD and then PD-L1. So I had to look that up. When I did, I found out that what you're dealing with here is a ligand and a receptor molecules for cell-mediated killing of, is this right? The tumor cells secrete one of these molecules, or has one of these molecules on its surface in overexpressed amounts.
Well, I think our clinicians
So-
will find it exciting that we're mentioning PD1 and PDL1 and PDL2.
'Cause they looked at that too, right?
There's a whole group of these programmed death receptors and ligands.
Programmed death, okay.
They're a big
Programmed-
deal.
They're
PD1
a
is
big
a big deal. Yeah.
And people who, maybe some of our readers, I think the New England Journal of Medicine in the last month, there was maybe one or two articles actually on blocking, targeting these. And there was a little editorial, which I'll have, Unleashing the Hounds. And when a person has a tumor, one of the reasons the tumors can succeed is that the immune system doesn't recognize and destroy it. And the tumors seem to have the ability actually to induce tolerance. And by interfering with this PD1, the programmed death pathway, you can in a sense unleash the hounds, the T cells that will target and destroy the tumor. And this was discovered decades ago, but now people are finally taking that basic science and trying it, and it seems to be exciting stuff. Seems
So
to
it
be
is,
working.
but they find that the secretions of this parasite, or the excretions of this parasite, actually upregulate PD/L1.
L1, so the ligand.
And so what is the significance of that?
Again, I think this feeds into the, just as a tumor does, you want to modulate, you want to not have an immune system that's destroying things that shouldn't be destroyed. If you're a tumor, you don't want to be destroyed. If you're a human being with insulin-producing cells, you don't want these cells going in and destroying your pancreatic.
So this prevents these
Destruction,
Okay.
By the way, Daniel, in a couple of weeks, Arlene Sharp, who works on PD1, she's at Harvard, she's going to be giving a seminar in the Wednesday microbiology series.
Great.
She'll probably, because she's
forward to that.
published
You get a
on
chance
PD1.
to ask her about fasciola.
I will not be talking to her, but
I bet she will not have read this article.
You don't
Maybe,
know. You
but
don't
maybe.
know. So the bottom line of the article's last paragraph in the
we're
discussion
at that time.
was the most telling, and that was that they addressed Vincent's question of what's in the super mix of what's doing all of this? And when they looked, they compared it to something called SEA. So that's called soluble egg antigen. So soluble egg antigen,
from
what is
another
that from?
parasite?
It's from schistosomes.
Schistosomes, yeah. And it's a less complex mixture, right?
Well, the fasciola is less complex than SEA.
Oh, SEA is more complex. Okay.
So SEA is derived from the eggs of schistosomes, and they modulate a whole bunch of responses in the liver.
And does it have a similar effect on type 1 diabetes?
It does.
In the nod mice?
It does. So they're now narrowing it down to just 2 molecules.
Who's going to win?
Maybe they both do.
Oh, by comparing the two, they can see what's in common?
Yeah, they did. They found 2 molecules that they both have in common. And so they're now busy doing experiments, seeing whether they can substitute just those 2 molecules for the either SEA or for the fasciola excreted proteins or whatever.
Daniel.
Yes.
What needs to be done before any of this gets into a human?
Great question.
No, I'd say a lot. This is still
it's still too early.
And one of the things we talked about was who do you give it to and when do you give it to them. So understanding when is that vulnerable initiation phase, and so I think there still is a lot, but we're moving in the right direction.
So let's say they identify a protein, right?
Right.
And this is going to be a worm protein.
Yes.
Right. Would that be a therapeutic, or are they going to then try and figure out what it does and make perhaps a small molecule that mimics
Vincent,
it? Right?
when you say the word protein and mammal, you're going to get an antibody
that's right.
to it, then eventually it'll be ineffective, right?
Right.
So you have to find out what it does and get a mimetic that's small enough to avoid an immune attack.
But in these mice, it didn't matter because, as Daniel said, it's one shot and it protects them for life.
Well, that's interesting, though.
So maybe
the life of a mouse isn't very long compared to a human, but they didn't go out to 2 years or a year and a half for these mice to see if they were still
30 weeks. 30 weeks.
30 weeks is that's less than a year.
Yeah.
But still, that's very, very suggestive that it does have a permanent effect, and that's remarkable, actually.
I mean, one of the positives might be that because this substance immunosuppresses, I'll say that, or induces regulation, it may actually be tolerated because of the action of itself.
Oh, that's what you mean.
The parasite is producing something that does not trigger an immune reaction. So we may not trigger an immune reaction to this substance, whatever it is.
That would be remarkable.
Now, the other problem is that this may have other consequences that are not obvious in this mouse, right? Sure. By inducing this pathway of macrophages and regulatory T cells and so forth, may make people terribly susceptible to some other infection or
tumors, right? As we talked about, this is what your tumors want to do. They want you to be very accepting. So and is there a critical?
So it sounds exciting, but there is a long way to go to make sure
you have to dampen the enthusiasm. Well, this.
No, you can be enthusiastic. That's good to be. Just don't inject it in people.
That's right.
Until we know more.
They're not injecting these stuff in people. They're giving them whole worm infections instead, and that's even more unacceptable because, to be honest with you, I would rather have a protein than a worm.
People will clamor for this. That's how it works.
I know that. So really, the review article that I snitched this chart from, this table, shows you all of the associations.
Even trichinella.
Well, that's what I'm going to tell you.
Dickson, maybe you could go back into business.
Oh, yeah, brother. Oh, it's been so long. When I was a graduate student, though, I did this following experiment, and in fact, that's probably the basis for my thesis. I took diffusion chambers, and I put millipore filters on the diffusion chambers of 0.45 micron pore size. And then I filled the chamber with the nutrient solution that I was maintaining the larva of trichinella in with trichinella larvae. Because what I really wanted to do was to control the exposure of the host to the secretions and excretions of living worms. I didn't want to take from the in vitro cultures. I wanted the living worms to actually produce what they could produce. And then I would withdraw the chambers from the peritoneal cavity at various times and then wait an appropriate amount of time and then challenge the mice to see whether they had gained enough immunity from exposure to become immune. And I found out that's trivial. But here's a way of giving somebody a worm infection without giving them a worm infection.
Yeah,
you could contain the worms in diffusion chambers, implant them. The worms will stay alive. They will not infect the host, but they will produce their secretions for a very, very long time. And in fact, you can actually prevent the immigration into the chamber of immune cells, keeping at bay the immune responses that might do harm for these parasites and keep them alive a very, very long time. I don't know if anybody's thought of treating like Crohn's disease.
Well, look, it looks like the trichinella is advanced quite a bit because they have specific effects of antigens on Th2 cells, FOXP3 regulatory T cells.
Yes, exactly. That's why.
So, and in fact, there's a whole list of, as you said, parasites here.
That's right.
And that's because these parasites need to manipulate or modulate their host.
Manipulate is a better word for
otherwise, they get eliminated. So
That's
we can take advantage of that.
Got it right.
As long as we can remove any of the negatives, right?
This is a very interesting potential sword into a plowshare.
And I wonder, actually, as we were talking about, when do you do this? When do you try to induce this tolerance? And I think with diabetes, it's tough because we're not sure.
Mm-hmm.
But I wonder more about transplant because we know when the problem with tolerance is. And these medicines are incredibly toxic and difficult. So maybe that's a
So what you're suggesting is if you're going to do an eyelid transplant, before that you would give whatever the active ingredient is here to not allow them to be destroyed.
Yeah,
right?
That's that would be potentially a way to.
Okay.
Well, because then we know when the autoimmune reaction.
This would work for cardiac transplant, also, and kidney transplant, and liver transplant, and lung transplant. It would work for any.
Well, it depends. Potentially,
it depends what the mechanism of rejection is.
And a lot of that work has already been done. It's buried in the literature, but it's out there, and so you can access that too. And so this is it's very exciting stuff.
So just to bring it to the virus side, did you know, Daniel, when you get a liver transplant, you get severely immunosuppressed with drugs, but then you have all these viruses in you that start to replicate and cause a problem because your immune system is shut off. Cytomegaloviruses and herpes viruses. We have to figure out how to make a less blunt, as you say, sword. Yeah.
Yes.
Here.
Cool. Anything else, Daniel?
No, but I'm excited to move on to our new mystery case.
Let's do it.
Oh, great.
Okay.
You have a lot of these.
You know what I did is
you brought us today,
I sat down and I made up a number of cases. So we have plenty for a while, and we'll have to figure out when we're off mic. We'll talk about in 2 weeks. I'm going to be down in the islands, not working, so we'll have to reschedule. So we'll either have to do this in a week or 3 weeks. So just our listeners maybe in suspense.
Give them a tough one then. Give them a tough one.
But let me present the case, and this is. I like this case. It's and I'm going to try to have Vincent and Dickson here get a little more of the history so they get it all. But there's an 18-year-old gentleman, and that's like at the lower age at which I deal with people. And this 18-year-old is seen in February. He comes in on a Thursday reporting 2 episodes of feeling poorly. He reports that on Monday, he initially started to feel poorly, and then developed shaking chills, followed by a high fever, very quick onset. 1 hour later, he goes to bed. 1 hour later, the fever passed, and he feels not quite so good until the next day. Next day, he feels completely fine, but then on Wednesday, the same things happen. He starts to feel not so good, shaking chills, very high fever, crawls into bed, a little bit of stomach discomfort, some nausea. He said that when he was having the fever, there was a headache. He just wanted no one to touch him or bother him. He's an 18-year-old boy, so probably that's because his mom is the only one who wants to come and comfort him. The following day, the patient schedules this doctor's appointment at the prompting of his mother, who says, this just doesn't seem quite right. Let's go see the doctor. And let me have you guys ask some questions at this point.
Brother.
It's very broad, right? 18-year-old kids.
Yeah, sure. 18-year-olds. Did he just get back from a tour of duty out in Afghanistan, or has he done any traveling?
So you want to know about travel?
I do.
He says, so here we are in February, and he did travel in the summer. He said he went on a trip back in the summer.
Where did he go?
Oh, he... I'm going to
jump in anytime, Vincent.
Jump anytime. He went to, it says here, Guatemala.
What?
He went there with it was a charity thing. It was very interesting. He went
Red flag goes up immediately.
He went there to help in the delivery of dental hygiene. Apparently there were 2 options with this group. His father's of Cuban descent, and it's this charity that goes down. You can either help with building latrines, or he can help with dental hygiene, which he explains is really distributing these fluoride packets and toothbrushes to people that have no teeth already. So he wasn't sure it was that.
How long was he down there, Daniel?
He was down there for a month. Down there for a month.
Okay.
Wow.
Other questions?
Is he chemically clean, as you would say?
No toxic habits.
No toxic habits.
That's true.
And was he...
No toxic habits.
Did he follow orders of someone that said, there are some infections that you can prevent or at least mollify by taking these things first?
He was told there was a risk of malaria down there.
Oh, good.
And he took his malaria prophylaxis.
Did he?
As prescribed,
and which
including
was
on
mefloquine?
return.
He took mefloquine. Was that his?
Yes, actually.
He took mefloquine.
Did he have any sex while he was there?
It doesn't mean that you're not going to catch malaria.
It has nothing to do with
He
my
might be
question.
thinking about other things.
I'm thinking of other things.
Oh, okay, fine.
Was he asked that?
Of course. And I think that's a good point. If you do any kind of travel or health to do with travel, people have more sex when they travel.
Really?
The amount of sex goes up, and it's one of these things that, particularly if you have an 18-year-old male who's going on a trip, you should probably tell him to bring condoms with you, even if you don't think you're going to use them, because there's a lot of diseases out there. And yes, this boy didn't actually.
Okay.
More questions.
And
Do you want to know about the setting of what it was like out there?
Where? Guatemala?
In Guatemala.
Sure.
Well, I can imagine, but go for it.
Because he sort of thought it was neat to tell me this whole story. So he travels there, and apparently there was a mix-up in where he was supposed to be, because originally you would go and you'd be in one place and you'd stay there, and that was the deal. But for him, they moved him around to a different site every week. And he was describing where he lived. And so he would be in these huts with dirt floors, and and he did bring mosquito nets, and he tried to sleep under the mosquito nets.
He took his mefloquine once a week.
He did take his mefloquine once a week. Actually,
That doesn't prevent malaria; it just prevents the disease.
And he was telling me that in a couple of the villages, there'd be his hut where they were staying, and then there'd be this river. You had to walk through this river, and he would take his shoes off and walk through the river barefoot.
There's no shoes
to get to where he's going to go, help the villagers, and then he put his shoes back on.
what did he drink?
Eat and drink. Yes.
What did he eat and drink? Well, the group took care of, made sure they did bottled water. And,
Okay.
All right. So
they also provided him with food as part of
no
their
unusual
group
eating
experience.
habits then.
He doesn't report any.
Right. And how long ago was that compared to when he got into the hospital? What's the
He was
incubation
in
period?
Guatemala in August,
Yes.
and he shows up in the hospital he's actually in the outpatient clinic. He's not in the hospital. This is
August?
February in the clinic.
February.
So he got August.
And that was the first time he's had these symptoms, right?
He was
Not
completely fine until February. Boom, he gets hit with this.
So this is going to be a malaria case, which you're leaning towards, at least thinking about.
before. No, he took his drugs, though.
He took his drugs. Well, I said it doesn't prevent infection.
So when he came back, I guess he
so this
stopped
could
taking
be a
his drugs, right?
Vivax-like infection.
When you come back, you stop taking your drugs, right?
You do. Well, you take them for a week afterwards, too. You take them a week before, weeks during, and then a week afterwards, and then that's it.
Or depending on the drugs, you might take them even longer. If it was a doxycycline or mefloquine, you might go longer.
Yeah, but
And his mom confirmed. She said, no, this is a good kid. He does what I tell him to. I
Right.
saw him take his medicine.
And he said he felt poorly, but can we get more specific? Did you say he had a fever?
Yes.
Did he have any joint aches?
The feeling poorly was this, we call it the influenza-like illness, right?
Right.
Everything hurt. I just don't feel right. I feel weak. I feel achy on my muscles. And then the pretty abrupt fever and the headache with the fever. And then I just want to go to bed. He goes to bed, and then an hour later, he's like, oh.
He's fine, and then it comes back.
He's fine, but he's still like a little bit, he's
dengue.
fine the next morning.
I'm also thinking of chick,
yeah, that's
rapid
That's
onset.
very true.
but it doesn't go away like that.
No.
Okay.
And by chick, I think he means chikungunya.
Chikungunya.
I believe he
One
was
of my favorite.
chicken gumbo
one of my
is my
favorite
favorite
diseases. If you can have favorite diseases.
Right. It's a new one in this hemisphere, yes.
Yeah, we have a number of cases imported into the U.S.
We do.
and it will be
So-
here endemic eventually.
What other questions could we possibly think of before Daniel rushes off to his seminar?
Well,
Do you remember how we did past medical history? We always ask that about people.
Yeah.
Sometimes that's helpful.
Yes, has he any unusual medical history?
Has he ever felt this way before?
Does he have a spleen?
He has a spleen, but no tonsils. He has some sort of colitis. He's had this for years, always, and he's not really sure at this point what it is. He's hepatitis B immune after natural infections. He's got serology that matches with that. He doesn't remember. He has thalassemia minor.
Really?
He was an Rh positive baby requiring transfusions at birth. He's not allergic to anything, and the thalassemia minor, as I mentioned, is part of the family history. He's a student. He's living with his parents in Flushing, Queens.
So thalassemia, major or minor, modifies the pathology of a typical malaria infection. So we can think about that as a way of saying, well, he got infected, but he didn't get sick right away because he has thalassemia, that sort of thing.
Okay.
That's one way of looking at this, of course, if it turns out to be malaria, but it doesn't have to be, of course.
And exposure history.
Exposure history. Bitten by mosquitoes repeatedly.
Yes, of course. And there was a lot of animals. All kinds of different animals. There were dogs.
Right.
There were chickens.
Right. Monkeys.
There were no sheep. No sheep. We're okay. People
But he
think
he didn't get bitten by any of these.
Cattle.
Not that
they
he
have cattle
reports,
down
no.
there, though. They do have cattle.
They do have cattle, and he's got dogs and cats in the home in Queens.
And they have vampire bats also.
Okay.
This is not a case of rabies, though. That's for sure.
He didn't eat any raw meat.
No.
So we're thinking about other vectors besides mosquitoes. What about little no-see-ums? Because they have leishmaniasis down there also.
Well, he reports that he didn't see any of those.
Perfect. That means he was bitten repeatedly. Half of those bites were from no-see-ums.
That's very good.
That's excellent.
All right, you have 5 minutes.
Okay.
And he has to leave. Anything else we want to know?
Vitals. He's not febrile when he sees you. He's looking
No
fine.
splenomegaly?
No, not that this clinician
No
could detect.
hepatomegaly?
No.
No megaly of any sort? No confounding physical
You're looking at the skin.
signs.
He looks fine.
It looks normal.
He looks fine.
And there's nothing else you're going to...
This is early onset.
And we do have some labs. I'm going to give you a few labs.
Wait, did he ever feel like this before? This is the first time ever?
He never felt like this way, ever before.
Okay.
But people tell you that all the time. I've never felt this.
How about his mother? Has he ever felt like this before?
No, but he did say that. I've never felt that sick before. When he gets this fever, he wants to go to bed. He's never felt that bad before.
Okay.
Labs.
Some more labs.
Do you want labs?
Yeah.
Sure.
I'm just going to give you a few labs.
Yeah.
Okay.
His white blood count is, with the CBC, he's got a decreased hematocrit. So we know what that is. The red cells are down.
Right.
His platelets are a little bit down.
Yeah.
His bilirubin's a little bit up.
That's a good
His
sign.
glucose is a little bit down.
Right.
And next week I'll tell you guys about more.
And the blood smear.
Are you going to want to? Everyone wants blood smears.
Well,
Dickson, why do you say it's good when he said those signs? Why did you
because
say
it means that the red cells are being destroyed and the bilirubin levels are going up because that's a byproduct of breakdown of hemoglobin.
Okay.
And it comes out in the urine.
And I don't want you to be my doctor if you're going to think that's good.
No, I didn't think it was good for the patient. It's good. It's a sign that the normal process is going.
It's good for you figuring out what's going on.
Yeah, that's right.
Oh, okay.
So maybe that is good for the patient.
Perhaps, yes,
You're giving
because
Dickson the right clues. You might actually treat him.
You wouldn't, but if you were forced to treat based on what you know only from this, well, how would you treat that patient? I mean, because in many places in the world, you don't have access to these kinds of tests. You see a patient, you make that decision, and you start treating.
Well, you treat based on what's
most
common,
common.
right?
That's right.
Yeah.
That's correct. So you're going to miss the Ebola outbreak. You're going to miss the Lassa fever outbreaks. You're going to miss.
Well, sometimes
you can't treat dengue, so that doesn't matter. So you start treating for what you can treat.
Right.
And so they start treating for malaria, probably.
Well, sometimes these Ebola, they're misdiagnosed as malaria, and they start treating for that. Right?
You remember how they found West Nile virus?
How?
No, they thought that was a clear-cut case of sleeping sickness, and of course it turned out not to be.
So, Daniel, I know you have to leave, but before you do...
Yes, there'll be the whole introduction, so I probably have about 3 or 4 minutes. Where they introduce the speaker.
Sure. We're going to do some email after he leaves, okay? Is that okay with you, Dickson?
Of course.
I just want to mention that you brought us both gifts.
Yes.
He came bearing gifts.
He did.
Do you want to describe
Beware
them,
of
Dickson?
Greeks bearing gifts.
We both have the same set. And Dickson, what do we get?
So Daniel has these rare abilities to penetrate into the inner soul of my passion. And what he did was he made a tie and a set of cufflinks for both of us. And he took an image from the TWiP icon, which is the icon for TWiP. It's the nurse cell photographed by Eric Gravé, and tiled it and he made a whole tie out of it. And the cufflinks also have an imprint of this. So we could become like the Three Musketeers, or
Do you have the same tie and cufflinks, Daniel?
Well, I'm having a bow tie made. That takes a little bit longer.
But of course it does.
Today your bow tie is the red ribbon of AIDS, right?
The AIDS awareness ribbon.
AIDS
yes.
awareness.
Right.
I
So
have it.
deeply appreciate the gift.
Thank you, Daniel.
Deeply. I
Sure.
will wear this.
Now let me just leave
with
you guys
pride.
with a couple. I know you're very excited about the concept of malaria. Does it bother you that it's February, that he was exposed potentially in August, and that he took his medicine just like he was asked to do?
No,
and
but
his
because I already
mom-
told you that no one believes that mefloquine prevents the infection.
Okay.
So let's say that it prevented let's say this person got infected with Plasmodium falciparum. Then he wouldn't have that because there's no liver stages that persist, and therefore the drug would wipe out the blood stages when it came out, and he would be cured. But if you're thinking about relapsing malaria, Plasmodium vivax, which is common, not as common as it used to be, but it's common, then this is a real possibility. It's still within the purview of a patient that has thalassemia minor. It modifies the pathology, and it stretches out I wouldn't call it pre-patent. This is pre-incubation period for the time when he starts to get sick from it. It could be a latent infection. He could have fought off the first round of parasitemia by the fact that he has thalassemia minor. He didn't get sick, but the parasite's latent in the liver tissue, waiting to come out at a prescribed period of time, and that as it comes out, every wave of infection has a different effect. And this particular one, it rose above the clinical horizon, as they would say. So that's, the one way to teach this to students is to point out the obvious first, but I know this is not going to be an obvious case because we've already had 2 obvious cases, and Daniel's probably working on a non-obvious case right now. So, I'm willing to think out the box and think about what else could this be if it wasn't malaria. That's the point. So if it's not malaria, what else could it be? And you mentioned some viral infections, and those were good choices, but didn't fit the clinical picture.
Doesn't. Nope.
So there's another parasite that does this, though.
Yeah, relapsing fever.
Correct. Borrelia recurrentis. So that's a TWiP organism. Could be that. I don't know what it looks like in thalassemia minor patients, but I have no data in my limited Rolodex.
What else causes relapsing fevers?
Oh.
Does treponema do that?
Treponema does that?
That's why I asked about sex, but apparently he's okay.
Okay, but there was no primary lesion?
Well, remember, he's an 18-year-old boy. He told me he didn't have sex.
That's true.
And his mom is there in the room while I'm asking him, of course.
It's funny.
Right.
It's funny.
Of course not.
There was an article in the Times.
Sex? What's that?
There was an article in the Times about sex and how hard it is to do surveys about sexual practices, and they gave an example of how often people have sex. It says, there aren't this many condoms sold in the U.S., so it can't possibly be.
That's right. It exceeded the limit by 10,
Jesus.
factor of 20.
Incredible.
That's right.
Yeah, so that's a
right.
good point. It may have
So there are plenty of unanswered questions here that need to be addressed, and you'll have fun with this, I'm sure.
Yeah, hopefully people will.
But right now your seminar is about to start.
I am off because it's Rafael Casalis, who is a B-cell.
yes, I saw that. Well, enjoy.
Genomicist.
And then I'm going to meet with him later, so it's always nice if you
Nice.
attend the
It
lecture.
would be
And yes,
almost
exactly.
essential.
Thank you, Daniel.
I would
See
say.
you,
Well,
see you
thank
next
you.
time.
Yeah.
Here you go.
Here we go.
You could read the first one. Would you like to do that?
I would
It's from
take great
Varun.
pleasure in reading the first email. Okay. Greetings, Provs.
Who's this from, Dickson?
It's from Varun. You said, would you like to
You
read
actually heard me?
it from Varun? Of course I
I'm
did.
surprised.
There's often a reference made to the connection between toxoplasma and schizophrenia, which has come up again in TWiP 79. There is a great deal of debate in the literature. I've also blogged about it some time ago. I have referenced Vincent's page in the Post. As someone currently doing some experiments in psychoneuroimmunology of schizophrenia, I am currently looking at it, keeping my fingers crossed, and then he gives a reference for his blog. Second point that came up in response to letters was regarding metallogens. As you know, there is a huge interest in the microbiome. And methanogen, I'm sorry, I said metallogens. I meant methanogens. And methanogens is also one of them researched upon. There is a correlation made between M. smithii and obesity. I have blogged about that also a time ago. I'm not aware of recent developments, though, and he gives a reference to that blog also. Last but not least, I enjoy Dickson's every episode. He had me read this on purpose. I didn't get down there until just now. He is an excellent science narrator. I, with thousands of others around the globe, just wish he keeps giving more shows and gets TWiP 100 within the end of 2015. As always, you probs are excellent educators. Thank you for your podcasts, Varun.
Varun's in Bangalore.
He's in Bangalore. I've been to Bangalore.
I think he's worried that you're going to depart.
I've been. I hope. Well, eventually we shall all depart, Vincent.
Yeah, but sooner than later. You always do that. And I can tell it's because you're insecure about it.
We will be staring at our eternities at some point in our lifespan.
I'm going to bug you in
You can do
the
whatever
afterlife.
you like. You know what? I don't think so.
I'm going to say, Dickson, are we doing a podcast or not?
Can you put this iPad on for me? If we do, then we'll have one.
Peter writes, Dear TWiP hosts, you get it? TWiP hosts.
Got it.
Congratulations to your new edition. I'm looking forward to future TWiPs with more clinical perspectives. Speaking of which, are you considering to get Alfred Sacchetti on TWiP? He's been on TWiV and TWiM, two of my most favorite episodes. It's been a while. I don't know if you remember, but he's a ER physician in New Jersey.
I do remember.
He's talked about infections. He's really fun.
Yep.
If there are not enough parasites in the ER to fill a show, maybe he could also return to TWiV and or TWiM. I really would like to listen to a show, Parasitism 911, if there's a next to talk about. And lastly, Duke University's course, Tropical Parasitology, Protozoans, Worms, Vectors, and Human Disease, starts on Coursera.
Nice.
I enrolled and hope to find the time to keep up with the course during the 8 weeks.
Oh, that's
Thanks
nice.
for all your work. Kind regards from Incheon, Korea.
Right. Incheon is where the airport is.
I'm going to ask him if he sees enough parasites. So he's in Camden, New Jersey, at a very large hospital. What do you think? Probably
I'd
no.
say no. But there is an emergency room doc here. His name is Josh Stillman.
Oh, he was in he was on the show once, wasn't he?
I know, and he has a long list of parasitic diseases that he has diagnosed right in the ER because people bring him specimens, and he's got the knowledge to actually.
Maybe we could get him on.
I would love to get him on.
Are you in touch with him?
ER docs are very difficult to get in touch with.
Okay.
They're ephemeral people.
Okay, read the next one,
I will.
Dickson.
Cindy writes, I am sitting here listening to the newest TWiP that happens to be, quotes, living in a wormy world, and watching tapeworm segments crawl out of my sleeping dog's bottom. Diploidium caninum. Fascinating. Definitely time to get to the vet. The dogs have been catching and eating rabbits lately. I'm thinking Taenia pisiformis. Well, maybe Diploidium. It depends. Sometimes I wish life wouldn't so closely imitate art. Anyway, thanks for the informative podcast. I think it is responsible for desensitizing me enough so I didn't lose my lunch. Twixt is reaching people like me, a non-scientist, stay-at-home mom who enjoys following science as a hobby, along with Skeptic's Guide to the Universe, which is fabulous. They are my most eagerly anticipated podcasts.
You've heard Skeptic's Guide to the Universe?
Yeah, I have actually. I've one of them, the Skeptic, has a wonderful website which I've referred to as a pick of the week.
How would a dog get a tapeworm eating a rabbit? What would that be?
Well, the intermediate stages are in the rabbit.
Which one?
Well, the
Taenia?
Yeah. But the other one, Diploidium,
mm-hmm.
you don't get by eating rabbits. You get it by eating a flea off the back of your own self. It contains the larva inside the flea because it ate an egg.
Got it.
So there are different life cycle stories here for the tapeworms, which are cestodes, by the way.
I think it's cool that we reach people like this who love
absolutely.
Science and
she
they
said,
look at things.
watching a tapeworm segment migrate out of the anus of her dog.
That's pretty weird. Yeah.
It is very weird.
Take her to the vet, right?
Actually, not so weird. It happens a lot.
Well, you got to give them something, right, Dickson? What would the vet give them?
You'll probably give them praziquantel.
Praziquantel. Steve writes, Hi, Vincent. No Dickson, no Daniel.
Well, I guess I'm not here.
First, let me say that I'm totally addicted to your podcasts and often find myself listening to them almost around the clock. I'm pretty much housebound with a long-term illness. And you and Dickson, and the team make a great contribution to keeping me the right side of sanity. You must be providing one of the best scientific educational resources on the web. Long may you continue. For want of science-literate doctors in the NHS, the National Health Service, that's the UK, right, Dickson?
It is.
I've been dumped in a pigeonhole, labeled either ME or hypochondria, depending on the preferences of the GP1Cs. I think that both are just handy labels that get the profession off from proper consideration of all the signs and symptoms, and that the majority of people like me would be properly diagnosed if only they could deal with scientists instead of GPs. I wish we had Daniel here. In this respect, I find myself quite envious of those who come to your research hospital and get the kind of careful consideration shown in your new case studies. If only there was somewhere like that here in England, but I'm afraid there is very much an anti-scientific, anti-testing culture in the medical profession here. The more clues in terms of signs and symptoms you provide, the more likely you are to be called a hypochondriac without considering them all. Anyhow, it's good to know somewhere there is a scientific approach. Because of this curious anti-science attitude here, a number of patients have actually started to conduct their own investigations, and I thought that Dickson in particular might like to look at the link below to some of the microscope work that one of the more gifted patients, Peter, has been doing. I have suggested to him before that he might find some people to help him at the AMA. I don't know if he did get in touch, but from the presentation below, I think you might agree that he's doing quite well on his own. Best wishes, Steve. He sends a link to a microscopy presentation.
Yep.
Which he just referenced. Borrelia spirochete microscopy. Wow, beautiful.
How about that?
Look at nice little
I'm going to
diagrams.
take time to look at that.
Very nice. And movies and so forth. So This is what you can do with a microscope and a little bit of patience, right, Dickson?
Absolutely. And maybe a lot of patience even.
All right, you're on
I'll
next.
read the next one. Russell writes, "Hi, doctors R and D and G."
RDG or DGR or GRD. Boy, you could make all kinds of...
You can keep going if you
3
want.
times 3 times 3,
How
right,
many
Dickson?
proteins can you make from triplets?
27 different combinations of 3 letters,
That's
right?
That's right. I've caught up to all the TWiPs, finally. Sorry, this may be a long email as I've had 80 episodes to think about. I have been listening to your show while living in Japan. I have been teaching English here since I graduated from undergrad with a degree in biochemistry. As an undergrad, I stumbled upon the opportunity to study parasites when I attended a professor's research presentation. The information was so fascinating and novel, I worked up the courage to ask, as a freshman, if there were any opportunities to work in our lab. Amazingly, I was accepted and will be forever grateful for the opportunity of the mentorship I received as a novice. I worked on characterizing trypanosome DNA binding proteins as therapeutic targets for the next 3 years. There I was, infected with an interest in parasites/infectious diseases. I decided to pursue a higher degree in the communication disease microbiology. In the communicable disease microbiology. I applied and was accepted to the University of Tokyo Graduate School of Medicine, International Health Sciences Master's Program. I'll be working on trypanosome mitochondria complex 2 as a chemotherapeutic target in the Department of Biochemical Chemistry.
Biomedical
Chemistry, sorry. Thank you for correcting me. The program starts April 2015, and so I have been following all of the TWiCs in order to keep my brain on point. I'm also reading a lot of related literature. I am now finishing Dr. Hotez's book, Forgotten People, Forgotten Diseases, which is an absolutely fantastic overview of NTDs and the public health issues from a drug development standpoint. I really hope that you guys have him on the show again. His journal, Plus NTDs, Neglected Tropical Diseases, is fantastic, as well as he regularly writes interesting public health review pieces on various topics. You guys have mentioned the Merguro Parasitological Museum in Tokyo. Yes, the only parasite museum in the world. I have been twice, and it's a great place. It's free, and they have great visual exhibits like phylogenite trees and many, many preserved samples, while most text is in Japanese. I was amazed to learn more about the huge impact Japanese researchers have had and continue to have in the field of parasitism and related fields. Finally, I must admit, I too was starting to lose interest in TWiP recently, but the addition of Dr. Griffin has re-energized in the broadcast. Dr. Griffin's expertise really brings out new expert participation from Dr. Despommier as well, whereas anecdotes' life cycles were starting to be a repeated lot. Maybe I felt I understood the life cycles and characterization of the main parasites well enough, therefore I was ready for more clinical experience to combine with them. Also a question. I missed some of the first TWiM and TWiV shows. Is there an easier way to get these earlier shows than going through the TWiV website and downloading from each episode page? Please continue to get knowledgeable guests and hosts on.
Please
Thanks.
continue to get.
I know what he meant to say. Thanks so much for your efforts,
A little
Russell.
snippy today, Dickson.
P.S. Well, you're picking. You're sniping. P.S. I wanted to add that I am an mid-20s American, and thus I appreciate the pun banter on this and all of the other Twixt broadcasts. Even when Dickson tries to stump Vincent with pop culture trivia, that seems to go well over my head.
What pop culture trivia do you ever stump me with?
None. You're way ahead of the curve.
Speak friend and enter. What's that from?
You're way over
You have no idea.
It's a quote from Star Wars.
Oh, it's close. Lord of the Rings. The Merguro Parasitological Museum. Yeah, I never went.
Great
I'd love to
stuff.
go, but have you been?
No, of course. I've never been to Japan.
I'm surprised. They have vertical farms there.
Never. I know. I'm dying to go.
Why don't you go?
Well, I think time and money has something to do with
Well,
it.
you're loaded. What are you waiting for?
What?
You're
I'm
loaded. This vertical farm business has made you rich. In fact, I don't know why you don't fund these podcasts.
Vincent, keep talking. I'm sure that people out there are laughing.
Robin, our friend, who sent 2 videos about those African buffaloes from last time. One of them attacking a lioness,
Very cool.
and another one is more of an informational video.
Imagine a buffalo attacking a lion rather than the other way.
So lions are not terribly fierce, are they?
Oh, no, they're very gentle. They're you could lay next down to them and look like Daniel in the lion's den. That is exactly what that's like, Vincent. He's lunch. He's a little bit of a snack for them. When they get hungry, they're
speaking
vicious.
of lunch, it's 12:17.
This is true.
You want to get some lunch, Dickson?
I do. I want to go out and get some lunch.
All right. Maybe we could eat and snipe some more.
Sure. It's all right.
I meant
like
I meant to answer Russell. It's like what?
It's like water
running off a duck's back. But you were going to say duck running off a water's back.
I was going to say
a
that.
water buffalo's
and then
back.
see if you could pick up on that.
You want to know how to download some of the earlier TWiM and TWiVs. So they're all on iTunes as far as I know. Some of the older TWiVs are not on iTunes, and that's something we're working on. It's just a technical issue that we have
But you've
to get
done a
through.
nice job, by the way. Vincent does all the web work on this, and so I think he deserves a huge amount of
No,
credit for the amount of time and
it's a
effort
labor
that he
of
puts
love.
into it. Because it looks professional, and you're not a professional.
Oh,
So he's done for TWiP, at least.
would you say I'm a professional virologist?
You've done a professional
Dixon?
job. You are a professional virologist, of
That's
course.
the one thing I'm a
That
professional.
does not mean that you can be a professional webmaster, but you are very close to being one of those people. So when you go to the TWiP website, at least, all the TWiPs are listed, and it's on just 2 pages.
Yeah, he doesn't want to. He doesn't want
to.
to download them one by one.
But you should do that for TWiV also, though, because
Russell,
they're not like that.
I do have it for TWiV also. I have a list on a page.
Well, then why doesn't he?
He doesn't want to download them on each page. You have to go to the page, download them. He wants them.
right.
So there are things called pod catchers. It's for your iPhone or Android device.
Okay. It's
These
like
are free apps that you can subscribe to a podcast and it will
Okay.
get all the episodes.
Oh, I see.
I use Downcast. Is that what it's called? Yes.
Downcast. No wonder
Downcast.
you're so gloomy every day.
Downcast. And there's another one called Overcast.
Outcast.
Overcast.
No, you use the Outcast.
Anyway, they're free. You can find them in the app stores, and they will allow you to get all the episodes, in fact, very
Yep.
easily. So check those out.
Yep.
Needless to say, this episode of TWiP can be found at iTunes, microbeworld.org/TWiP, and on your favorite podcatcher, like Downcast or Overcast. And it is an overcast day today, isn't it?
We didn't mention the weather we don't usually do that on TWiP, but
Dickson, we started the show by talking about the temperature.
Yeah.
You don't even remember 90 minutes ago, do you?
We didn't say what the weather was like, actually.
We said
On
the
TWiP,
temperature
we give
was
a complete meteorological rundown.
just at freezing, but it's cloudy and overcast.
Yeah,
it's supposed to
dreary
snow
day.
tomorrow, I think.
Yeah, it's winter. What do you expect?
If you do have questions and comments, send them to TWiP at twiv.tv. And Daniel Griffin, of course, is here at Columbia University Medical Center, where he's some sort of staff scientist, right?
He's on board for being a clinical associate.
He's going to soon see patients, right?
He does now, but not here. He does that on Long Island.
In Long Island, but soon he will do it here, right?
Well, we're going to ask him to develop a website for clinicians and parasitic diseases. So maybe
he's going to develop a website?
Why not?
You don't think he has time to develop a website?
No, he might like to do that.
You think it takes no time at all to do this?
No, Dickson? But he might know a 12-year-old kid that can help him.
That's your downfall, thinking that 12-year-old kid can do that. Yeah,
I've had no downfalls, thank you.
Well, it will be your downfall.
But I do fall down a lot.
Dickson Despommier is at Racaniello.org.
He is.
Thank you, Dickson.
You're welcome, Vincent.
And I'm Vincent Racaniello. You can find me at virology.ws. The music you hear at the beginning and end of TWiP is composed and performed by Ronald Jenkins. If you like his work, go over to ronaldjenkins.com and check it out. He's a cool guy. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.